Welcome to the video about nucleic acids. Before we get started, I just want to let you guys know that nucleic acids make me feel hopefully you guys will feel the same way as I do about nucleic acids after this video. So, there are four basic nucleic acids. There are the pyrimidines and the purines. The purines are bigger than the pyrimidines. So let's look at the purines first. So the purines are adenine, and guanine. So adenine looks like this. You will have to memorize these structures, because in the next slide, after we detail what these look like, we will then talk about how they hydrogen bond to each other. So there's adenine, and what we'll see is guanine looks very similar, but yet different. What you can also start seeing is these are called purines because of their ring structure, and you should also start to see how they would maybe start to hydrogen bond with their pyrimidine copartner. So what's important to note, we do not find just nucleic acids, at least when we talk about DNA or RNA, just like this. They're attached to a sugar. These nitrogens are important for that connection. And so this is where we will see then the ribose connected. And we'll get into a little bit more of what that would look like in class. But for now, just we do need to memorize just the basic structures of what a nucleic acid looks like. So those are the purines. Now let's talk about the pyrimidines. So a pyrimidine is just a single ring structure. So we have cytosine, we have thymine, and we also have an extra pyrimidine, uracil. So cytosine looks like this. And again, we'll see very similar structures of the pyrimidines. And on an exam or on a quiz, if you don't like drawing every atom out and you just want to do the skeletal figure, by all means feel free to. So, then the last is uracil. So you guys probably know that thymine and cytosine are found in DNA, uracil and cytosine are found in RNA. Before we get to that, we'll talk about, here are the nitrogens that will again attach to either the ribose or the deoxyribose to make the nucleotides. So in DNA, we see thymine. In RNA, we see uracil. And the question is, why? We're not going to show mechanism here, but what you see is that cytosine can undergo a deamination reaction. And so when cytosine deaminates, so that, like I said, we're not going to talk about how mechanistically. It does take in water, and you kick ammonia. When that happens, things get rearranged, and you look like this. And so the reason why we use thiamine instead of uracil in our DNA is for when the natural deamination of cytosine occurs, our proofreaders can see we have a uracil in our DNA. That cannot happen. We need to do something about it. Whereas if we had uracil naturally in our DNA, we couldn't distinguish between the two. So, from those 4 bases, we know that nucleic acids can base pair together. And so let's look at a few of those base pairs. The first one I'm going to draw is between G and C. So let's draw a G. And so the thing that holds 2 nucleic acids together, or 2, I should say, nucleotides together in DNA are hydrogen bonding. So what we're going to see is a lot of hydrogen bonds. I'll just put sugar here. So this is the G. And then we'll draw the C. So alongside knowing the nucleic acids, you also should know how they hydrogen bond. So you will not only have to know what they look like, but you will have to know these base pairs as well. And so there's a guanine, there's a cytosine, and what you can see, there's a hydrogen bond formed between those 2 groups. There's a hydrogen bond formed between those groups, and then there's a hydrogen bond formed between one of the hydrogens from that amino group to the carbonyl. And so we know that G hydrogen bonds 3 times with C. So a GC base pair is a lot stronger than an AT base pair. And the reason is because of the number of hydrogen bonds. What we also see is the directionality that nucleic acids have. So when we draw these, we're drawing them from a 5' to a 3' direction. The other thing that we see is the major and the minor groove. And so the major groove is up here, and the minor groove is down here. And so what we see is the major groove has a little bit more space than the minor groove. a little bit more. Major groups are where replication enzymes, the majority of enzymes that bind DNA bind to the major groove because there's a lot more space. It's a lot more open. So that's the first base pair that we're going to talk about, guanine and cytosine. The next one we're going to talk about is adenine and thymine. This is also the same. We could also supplant uracil in here if we wanted to. So let's draw adenine. Oops. Let's redraw adenine. And what we'll see is the difference between G's and C's and A's and T's. So there's adenine, poorly drawn. And so let's put an N. And now let's draw thymine. So here's thymine. So we have adenine and thymine. What we can see is there will be a hydrogen bond formed between these two groups, hydrogen bond formed between these two groups, but there is no hydrogen bond before the carbonyl group and that hydrogen off a carbon. So those won't hydrogen bond. And so what happens is A's and T's only get 2 H bonds compared to the G's and C's, which have 3. So A's and T's are a little bit weaker of an interaction than G's and C's. There's one more type of base pair I want you guys to learn. This is not found in your book. It's known as the GU wobble pair. And so there are times when base pair, or when nucleic acids can base pair together in the non-Watson-Crick base pair, right? A's with T's, G's with C's. And this is very important when we start talking about structures of RNA, because these are the types of interactions that stabilize that structure. And so it's called a GU wobble. So obviously it's RNA because of the uracil. So let's draw guanine first. And what we'll see is that it's not a direct face-on-face or two faces attacking each other, but it is shifted, which is why we call it a wobble. So there's guanine. So let's draw uracil. And so the way a GU wobble would occur would be due to secondary structure or even tertiary structure of an RNA. So how the RNA would fold and how the RNA combines. In class we'll talk about RNA structure and different structures of nucleic acids. But this is what stabilizes them. So now we have 2 hydrogen bonds to stabilize. So it's the same strength as an A and a T, but it's between a G and a U, 2 things that we normally wouldn't think about bonding. So the question is, why do nucleic acids adopt a double helix? So if you look at a nucleic acid, let's just look at the pyrimidines just because they're easier and quick to draw. So let's look at just cytosine and we can see so what we can see, especially and when we see this or when we see the purines, what we can do is we can do some manipulation of the enzyme, or of the electrons, and what we can actually do is do something like, let's say, that. Right? So minus on the oxygen, plus on the nitrogen. So we'll talk about the different forms that these can take in solutions in class. But just the key to this is that the conjugated double bonds that we see in these nucleic acids. Look at your purines, look at your pyrimidines, make sure you can see it. Things that have conjugated double bonds lay flat. Flat as a pancake. And actually, one of the things when we talk about the double helix is why this flat like a pancake is actually very, very important. So, if these bases lay flat like a pancake, then why is there a twist and a turn in a double helix, and why does it not just look like this? Why does it not just look like a ladder? Why does it look like a double helix? Well, we have to think about what makes these nucleic acids, and specifically the nucleotides. So remember, a nucleotide is a nucleic acid plus its sugar plus its phosphate. So one of the reasons why we put a kink or we put a turn in our double helix is because of the phosphate. Those phosphates are negatively charged. And when you get two phosphates close to each other, they want to repel each other. And as they're repelling, that causes the double helix to twist. The same thing, so that's the first reason, is the negatively charged phosphates. The second reason is the sugar. So I didn't have you guys memorize the chair structure, but if you go and you look at way sugars actually look, they're big and bulky, and they have a little bend to them. And so to make sure they have enough space, the nucleic acid has to adopt a double helix where it can turn. In class we'll talk about the different types of double helices that DNA can adopt. So DNA is very stable. DNA is our genetic code for a reason. However, we can still damage our DNA. One of the ways we can damage our DNA is UV light. And so let's say it's a poorly drawn sugar. Oops, phosphate. Connected to another sugar. So those are just a representation of a sugar. And let's say we have cytosines, one on top of each other. Or we can also have thiamines, one on top of each other. Now, one of the things we said is that these things are flat like pancakes. And one of the things that stabilizes these interactions are some van der Waals interactions between the top and the bottom, right? Because if you have conjugated double bonds, there is some electron density above and below it. And so those two things interact, stabilizing the double helix. So let's say we have this. And then let's say you go out in the sun and you get a suntan, or you go to the tanning bed, and then let's say some UV light gets into your DNA. And you see a cytosine above a cytosine, or a thymine above a thymine. So usually pyrimidines on top of each other. What you can do is you can actually cross-link those two pyrimidines. They're known as pyrimidine dimers. So pyrimidine dimers are very bad. They cause this huge lesion or this huge bulge in your DNA, and they put a kink in your DNA. We obviously have repair mechanisms to do this, or to help out with this. We're not going to talk about them here. That's for advanced biochemistry if you would like. UV light, that's one way we can damage our DNA. The second way we can damage our DNA is through radiation. So if you've ever gotten an X-ray or another type of treatment with radiation, they usually protect other parts of your body from that radiation. And here's the reason why. So we have our piece of DNA. I'm just drawing them connected. We can think of radiation as a bowling ball. And what we're doing, whether it's a neutron or a variety of other things, it's at a high rate of speed. We throw that bowling ball right at our DNA. And what happens is we make a double-stranded break in our DNA. Double-stranded breaks in DNA are very bad for organisms. They cannot fix them right away, or when they do fix them, they are very error-prone. And so too much radiation destroys too much of your DNA, and that's why radiation is very poisonous. The last thing we can do to damage our DNA is use a chemical called EMS, or ethyl methane sulfonate. So we do this in the lab to make more targeted mutations in the DNA. So here's what this chemical looks like. We're not going to talk about the mechanism of how this works. We just have to know that it turns G's into A's. Right? So instead of a GC pair, it makes AT pairs. And it has a mutation rate of about 5 times 10 to the negative 4th to 5 times 10 to the negative 2nd bases per gene. So it's actually very good at what it does. And so we use it in the lab to make random mutations in a bunch of different genes, and we look for our gene of interest. So, I told you guys there are 5 main bases, right? Adenine, guanine, cytosine, thymine, uracil. There is a 5th base, or at least we can modify bases, and then they get modified enough and we use them so much because they mean so much to us that we call it an extra base. So this is technically the 5th base because we look at thymine and uracil as interchangeable. So let's draw cytosine. So there's cytosine as we know it. We know that the 5th position of cytosine can be methylated. This is known as 5-methyl cytosine, or 5-MC. Right? So 5-methyl cytosine plays a huge role in our bodies, in our gene expression. Whoops, I forgot that. In everything that we do. So we'll talk about this more in class, but I just wanted to bring this up, the structure up to you. If you have any questions about it, please come see me. Contains your genetic code in a molecule that makes up all your chromosomes. Double helix structure, a two-stranded spiral. Set up instructions for your cells so that they know what to do, like send their signals and make new bonds. Make it proteins, one acid.